Introduction. Each chapter of the study guide consists of 5 sections: narrative, workbook questions, challenge questions, sample test questions, and other sources of information. In addition, each chapter includes a list of objectives and a list of the key terms introduced. Before beginning to read the narrative section, review the objectives and look over the list of key terms. Doing so provides a focus and orients you toward the concepts that you should learn in that chapter. The next step is to read through the narrative section. The purpose of this section is to serve as a primer and provide the basic concepts of the topic. Many illustrations, photographs, and charts are included to help you understand the material. The workbook section follows the narrative. The questions in this section are designed to reinforce the basic concepts presented and to review the terminology presented. These short answer questions help you determine which parts of the chapter require more study. Once you have successfully completed the workbook section, you should proceed to the section containing other sources of information. These materials provide greater depth and detail on each subject and help you apply the basic concepts to field situations. The goal of the challenge questions is to strengthen your understanding of the subject areas and help you apply the concepts learned. These questions go beyond the scope of the narrative. To answer them, you must demonstrate a more detailed understanding of the subject and be able to project the concepts into practical applications. The final section is the sample test questions. These questions are typical of those that might appear on the certification exam. Their purpose is to help familiarize you with the style and scope of the exam. The questions on the certification exam may correspond to, but will not limited to, the material presented in the study guide. Some exam questions are based on the other sources of information provided. Other questions reflect knowledge that an arborist would be expected to gain from experience. Many of the questions test your ability to apply the concepts learned in the study guide. Chapter 1. Tree Biology. Objectives. 1. Explain the structures and functions of the buds, leaves, wood, and roots of a tree. 2. Describe the interaction of structure and function in tree biology. 3. Describe the basic composition of a tree's vascular system and explain how water, minerals, carbohydrates, and plant growth regulators are transported within this system. 4. Describe the relationship roots have with mycorrhizae. Explain how the soil environment affects root growth and distribution. 5. Discuss the processes of photosynthesis and respiration and the factors affecting these processes. 6. Discuss how the growth and development of a tree is the result of the interaction between its genetic potential and the environmental surroundings. 7. Explain the concept of compartmentalization of decay in trees, CODIT. 8. Describe the basic anatomy and physiology of palms, including how they grow. Introduction. Trees, by most definitions, are large, woody plants with a single main trunk. To paraphrase Alex Shigo, trees are woody, long-lived, compartmentalizing perennials. This series of unique characteristics has allowed trees to dominate the vegetation of large areas of the world. Arboriculture is both an art and a science. It combines skill and craft with knowledge and fact. Thus, a foundation for the practice of arboriculture is a thorough understanding of how trees grow. The skilled arborist learns how a tree grows and how to care for and manage it in a way that supports its growth and development. Like physicians, arborists use knowledge of tree growth and development to diagnose problems, assess potential, and prescribe treatments. And, just as in medicine, prescription before diagnosis is malpractice, the arborist must understand tree biology before embarking on a program of care. The study of tree biology is the study of structure and function and the relationship between them. Anatomy and morphology are the studies of the component parts of a tree. Physiology is the study of the biological, physical, and chemical processes within these components, providing the basis for function. This text will focus primarily on the biology of hardwood trees, but some attention will be given to conifers and palms. Tree anatomy. Basic structure, cells and tissues. All living organisms share a basic organizational theme based on cells, tissues, and organs. Cells are the basic building blocks of structure. New cells arise from the division of existing cells. In trees, this process occurs in specialized zones, called meristems. Following division, cells undergo differentiation, which changes their structure and permits them to assume a variety of specific functions. Cells with similar structure and function are arranged into tissues such as bark or wood. Tissues are then organized into organs, of which plants have 5: leaves, stems, roots, flowers, and fruit. Finally, organs are organized into intact, fully functional organisms. Trees. There are 2 basic types of meristems: 1, primary, which produce the cells that result in elongation of shoots and roots, and 2, secondary, also known as lateral meristems, which produce cells that result in increases in diameter. Growth of shoots and roots from the apical meristems is called primary growth. The presence of secondary meristems, growing within the stems, branches, and roots and producing wood, allows trees to grow so large. The exceptions to this pattern are palms, which lack secondary growth. Meristems located at the ends of shoots and roots are primary, or apical, meristems. In shoots, they are found inside buds. The overlapping scales or modified leaves of buds protect both the meristematic region and the developing shoot. In roots, the apical meristem is protected by a root cap. Trees have 2 secondary, or lateral, meristems. The first lateral meristem is the cambium. The cambium is a thin sheath of dividing cells that produces the cells that will become the vascular system of the tree. The cambium produces 2 kinds of tissue, xylem to the inside and phloem to the outside. The second lateral meristem is the cork cambium, which produces the bark, periderm. Unlike animal cells, plant cells have cell walls. The structural component of the primary cell wall is cellulose, the most common organic compound on Earth. Cellulose plays a large role in providing the architecture of different types of cells. Lignin is another component of plant cells, and, when formed in the cell walls of the wood, it provides the strength and rigidity that allows trees to grow tall. Xylem and Phloem. The xylem, or wood of the tree, has 4 primary functions. 1. Conduction of water and dissolved minerals, elements. 2. Support of the weight of the tree. 3. Storage of carbohydrate reserves, and 4. Defense against the spread of disease and decay. Xylem is a complex tissue, composed of both dead and living cells. The living tissue of the tree is known as the symplasm, and the non-living tissue, which in mature trees represents a large portion of the volume, is called the apoplasm. Xylem of gymnosperms, for example, pines and spruces, is composed of tracheids, fibers, and parenchyma cells. Tracheids, which conduct water and provide mechanical support, are elongated, dead cells with pointy ends and thickened walls. Fibers provide mechanical strength. Parenchyma cells are living cells interspersed among the other xylem cells. Parenchyma cells located in the outer layers of xylem store carbohydrates, help defend against decay, and have a structural function across the grain. The xylem of hardwood trees is also made up of tracheids, fibers, and parenchyma cells, as well as vessels. Vessels are the primary conducting elements in hardwoods. The vessels can be thought of as stacks of dead, hollow cells that form long tubes of water-conducting elements. Vessels are much more efficient in water conduction than are tracheids. Another differentiating factor is that parenchyma cells are more abundant in hardwood trees and are arranged close to the vessel elements. The physical and biological properties of different types of trees are related to the arrangement of the cell types within the xylem. Some trees form wide vessels early in the growing season and narrower vessels later in the season. These trees are said to be ring porous, and they include elm, ulmus, oak, quercus, and ash, fraxinus, among others. Other species produce vessels of uniform size throughout the growing season and are called diffuse porous. Examples include maple, acer, plane tree, platymus, poplar, populus, and others. When a tree has been cut and can be viewed in cross-section, growth rings are visible in the xylem. These rings are the result of seasonal production of xylem by the cambium. They appear as rings because the relative size and density of the vascular tissues change throughout the growing season. As the season progresses, cells become smaller in diameter. Thus, the contrast between cells produced early in the season, early wood, and those produced later, late wood, allows the diameter increase within an individual year to be seen. As with most things in nature, there are exceptions. Conifers do not have vessels and are said to have non-porous wood. The growth rings of tropical trees, if present, may not be annual due to their constant growing conditions, and palms do not form rings of xylem at all. Not all of the conducting elements in the xylem transport water. In conifers, 2 to 12 rings may actually conduct water. In trees such as elm, only the outermost 1 or 2 rings do so. Xylem that conducts water is called sapwood. There are many living parenchyma cells in the sapwood. Farther inside the tree is the heartwood non-conducting tissue that contains no living cells and is sometimes darker in color than the sapwood. The phloem moves carbohydrates, sugars, produced in the leaves throughout the plant for storage or consumption. Movement of sugar in the phloem is relatively slow and occurs along pressure gradients. Phloem transport requires energy. Unlike the hollow vessel elements and tracheids of the xylem, the functional phloem is composed primarily of living cell sieve cells in conifers, and sieve tube elements and companion cells in hardwoods, and growing tissues, originating with the first year's shoot and remaining suppressed beneath the bark until growth is triggered. When dormant buds elongate and produce shoots, these shoots are termed epicormic. A node is a slightly enlarged portion of the twig where leaves and buds arise. The internode is the area between the nodes. Leaf scars and terminal bud scale scars are visible on new twigs and are useful in measuring annual twig elongation in many species. On the outer surface of the twig, lenticels permit the exchange of gases. Each branch of the tree is autonomous, producing and storing enough carbohydrates to sustain itself and exporting some to the trunk and roots. Branches generally do not import carbohydrates from other parts of the tree. Each branch is similar in structure and function to the entire tree crown, yet branches are not simply outgrowths of the trunk. Instead, branches and trunks have a unique attachment form that is critical to the application of arboricultural practices, such as pruning. Branches are strongly attached to the wood and bark below the branch, but weakly attached to the wood and bark above the branch. The annual production of layers of tissue at the junction of the branch to the stem is seen as a shoulder or bulge around the branch base, called the branch collar. In the crotch, the branch and trunk expand against each other. As a result, bark is pushed up to form the branch-bark ridge. If bark in the crotch is surrounded by wood, it is called included bark. Included bark further weakens the crotch because the normal branch-to-trunk attachment is not formed. Leaves. Leaves are the food producers of the tree. Leaves have cells with chloroplasts that contain a green pigment called chlorophyll. Chlorophyll is the primary leaf pigment that absorbs sunlight. The energy of the sunlight is collected in the chloroplasts where, in a reaction called photosynthesis, it is converted to chemical energy in the form of carbohydrates. A second role of leaves is transpiration. Transpiration is the loss of water through the foliage in the form of water vapor, which helps cool the leaf and draw water up through the xylem. The structure of leaves is uniquely adapted to carry out the roles of photosynthesis and transpiration. Leaf blades provide a large surface area for the absorption of sunlight and carbon dioxide needed for photosynthesis. Because leaves are thin, no cells are far from the surface. This structure facilitates the exchange of gases and absorption of light. The outer surface of a leaf is covered by a waxy layer called the cuticle. The cuticle functions to minimize desiccation, drying out, of the leaf. Stomata, small openings in the leaf surface, control the loss of water vapor and the exchange of gases. Carbon dioxide is absorbed into the leaf, while oxygen and water vapor are released. Trees regulate carbon dioxide input and water vapor output through the guard cells, which regulate the opening and closing of the stomata in response to environmental stimuli such as light, temperature, and humidity. Leaves have a network of conducting tissues comprising the veins, or vascular bundles. These veins are composed of both phloem and xylem tissue. They transport water and essential elements, and they carry carbohydrates produced in the leaf cells to other parts of the tree. Trees that shed their leaves periodically, typically every year, are called deciduous. Trees that hold their leaves for more than 1 year are called evergreen. Deciduous trees generally lose their leaves in response to periodic environmental changes, such as day length, temperature, and/or rainfall, as a result of cell changes and growth regulators that combine to form an abscission zone at the base of the leaf stalk or petiole. The abscission zone has 2 functions. 1. To enable leaf drop and 2. To protect the region of the stem from which the leaf has fallen against desiccation and pathogen entry. Fall foliage color in deciduous trees results from the breakdown of chlorophyll and the expression of other pigments contained in the leaf. Short, sunny days combined with cold nights enhance the accumulation of sugars and trigger a decrease in chlorophyll production. This process allows other pigments, including anthocyanins, reds and purples, and carotenoids, yellows, oranges, and reds, to be unmasked. Roots. The roots of trees serve 4 primary functions: anchorage, storage, absorption, and conduction. Larger roots are similar to the trunk and branches in structure. The main functions of large roots are anchorage, storage, and conduction. Absorbing roots are the small, fibrous, primary tissues that grow at the ends of and along the main, woody roots. The absorbing roots have epidermal cells that may be modified into root hairs, which aid in the uptake of water and minerals. As with shoot tips, root tips contain a meristematic zone where the cells divide and elongate. Roots grow where moisture and oxygen are available. Most absorbing roots are found in the upper 12 inches, 30 centimeters, of soil. Horizontal lateral roots are also usually near the soil surface. Some trees form sinker roots, which grow vertically downward off the lateral roots, providing anchorage and increasing the depth of soil exploited by the root system. The downward-growing taproot of young trees is usually choked out by expansion of roots around it or is diverted from its downward growth by unfavorable growing conditions. Few mature trees have taproots. The area where the roots join the main stem is known as the root crown. From there the roots spread out and decrease rather quickly in diameter in the zone of rapid taper to long, spreading, branching roots, 1 to 2 inches, 2.5 to 5 centimeters, in diameter. Roots may extend laterally for considerable distances, depending on the tree and soil conditions. Roots of trees grown in the open often extend 2 to 3 times the radius of the crown. The extent and direction of root growth is more a function of environment than genetics. Many roots live in a symbiotic relationship with certain fungi. The result of the association is termed mycorrhizae, fungus roots. In this form of symbiosis, both organisms, the tree and the fungus, in this case, benefit from the living arrangement. The fungi derive nourishment from the roots of the tree. In turn, the fungi aid the roots in the absorption of water and essential mineral elements. Tree physiology. Photosynthesis. Photosynthesis is the process by which green plants use light energy to build carbon molecules that make carbohydrates. Literally, photosynthesis means putting together with light. Photosynthesis takes place within cells that contain chloroplasts. Chloroplasts contain molecules of chlorophyll, the light-absorbing pigment that gives plants their green color. The raw materials necessary for photosynthesis are carbon dioxide and water. The tree absorbs carbon dioxide from the atmosphere through the stomata in the leaves. Light energy is collected in the chloroplasts. The light energy is converted to chemical energy, carbohydrates, and used for growth and development or stored as starch for later uses. Oxygen, the byproduct of photosynthesis, is released through the stomata. The carbohydrates of photosynthesis are sometimes referred to as photosynthate. Photosynthates are the building blocks for many other compounds required by the plant. Proteins, starch, fat, growth regulators, amino acids, and other important compounds are produced from photosynthate when combined with other essential elements such as nitrogen, potassium, sulfur, and iron. Much of the photosynthate is stored by the tree in the form of starch for later energy requirements. Respiration. Respiration is the process by which the carbohydrates generated by photosynthesis and stored as starch are used by the tree. Carbohydrates are actually chains of carbon, hydrogen, and oxygen molecules chemically bonded together. When the bonds are broken, energy is released, and carbon dioxide and water are given off. The tree uses the released energy for all biological functions. Both plants and animals respire. Respiration is a constant process of converting food into energy. Plants produce their own food, however. Thus, it is important that overall photosynthesis, food making, exceeds respiration, food using. When respiration takes place in the absence of photosynthesis, the tree must rely on stored energy reserves. If this occurs over a long period of time, the tree will eventually copyright 2010 by International Society of Arboriculture. All rights reserved. Figure 1.17. This diagram illustrates, in a simple way, the chemical processes of photosynthesis and respiration. Run out of energy and die. A practical example is a tree that is repeatedly defoliated. Without foliage, photosynthesis stops, and the tree cannot produce carbohydrates and store them as starch. Yet as long as the tree has living cells, respiration takes place, and energy is consumed. Oxygen is required for normal, aerobic respiration. Trees are incapable of respiration in the absence of free oxygen, anaerobic, for long. Under flooded conditions or with severe soil compaction, oxygen is scarce and respiration uses food at a much higher than normal rate. If these conditions persist, roots will die. Transpiration. Transpiration is the loss of water from leaf surfaces in the form of water vapor. The evaporation of water cools the leaves and creates a transpirational pull that moves water up through the xylem. The cuticle helps prevent uncontrolled water loss from epidermal cells on the leaf surface. Gas exchange, where water vapor and oxygen are released and carbon dioxide is absorbed, takes place through stomata, which are located primarily on the underside of the leaves. Each stomatal pore is lined by two guard cells that regulate the opening and closing of the stomate. Stomatal opening is influenced by environmental conditions such as light, temperature, and humidity. In trees, stomates usually open in the light and close in the dark. Temperature, humidity, and available water affect the rate of transpiration. Transpirational water loss is also affected by anatomical features such as cuticle thickness, the presence of hairs on the leaf surface, and the number and location of stomata. Some plants with a thick cuticle, small leaves, and sunken stomata are adapted to hot and dry conditions. Arborists sometimes use anti-transparent sprays to close stomatal pores artificially or to add an impervious coating on the leaf surface, thus reducing water loss during transplanting or under drought conditions. Use of these materials, however, reduces evaporative cooling of leaves, reduces uptake of carbon dioxide, and can reduce photosynthesis, absorption, translocation, and the vascular system. Water is essential for all living cells. Most biological reactions, including photosynthesis, require water. Water maintains cell turgidity and is necessary to transport essential elements within the xylem. Water and mineral elements are absorbed from the soil by the roots. The tree uses some of this water for growth and metabolism, but most water is lost through transpiration. This water loss creates the transpirational pull that helps move water through the xylem. The xylem can be thought of as a continuous column of water, where the evaporation of water molecules from the leaves pulls water up through the tree. Other forces, adhesion, cohesion, and pressure differentials, also contribute to the mass movement of water in the xylem, although the process is not fully understood. Water enters young roots or mycorrhizal roots, in part, by a process called osmosis. Osmosis is the movement of water through a membrane from a region of high potential, water concentration, to a region of low water potential. Pure water has the highest potential, adding anything such as minerals or sugar lowers the potential. Water normally moves into roots where the water potential is lower than in the surrounding soil. If the water potential is lower in the soil than the root cells, water will actually move out of the roots into the soil. An example is when salt concentrations are high in the soil, such as from de-icing or excessive fertilizer application. Phloem transport is the movement of photosynthate and other compounds in the phloem. The phloem is composed of sieve cells in gymnosperms and sieve tube elements and companion cells in angiosperms. Carbohydrates are actively pumped through the phloem, a process that requires energy. In discussing phloem transport, the terms source and sink are often used. Leaves are the source of photosynthate. The photosynthate moves through the phloem in a direction from source to sink, that is, from areas high in carbohydrate concentration to areas where more is required. Sinks are plant parts that use more energy than they produce. Almost all plant parts, including young leaves, are sinks at some time. It is sometimes thought that carbohydrates are produced in the leaves and transported, through the phloem, exclusively to the roots for storage. Most photosynthate is either utilized or stored in proximity to where it is manufactured, although it actually can move in either direction in the phloem. The movement of water in the xylem and photosynthate in the phloem are examples of longitudinal, or axial, transport. Radial transport is the horizontal movement of water or nutrients within the tree between cells of different ages, primarily through ray cells. Rays are living channels of cells through which water, elements, and carbohydrates move laterally. Control of growth and development. The growth and development of a tree is the result of the interaction between its genetic potential and the surrounding environmental conditions. There are many examples of this interaction. In nature, sweet gum, Liquidambar styrosiflua, may attain a height of 150 to 175 feet, 46 to 53 m. Yet rare ly does the species grow that large in urban areas. Although sweet gum may have the genetic potential to grow 150 feet tall, the urban environment limits the expression of that potential. Other genetic components work similarly. For example, the range of size, fall color, and form seen in many cultivars of red maple, Acer rubrum, represent the breadth of genetic potential within the species. Yet the red maple cultivar Red Sunset will never look like buds on the same shoot. Strong dominance is confined primarily to the current season's shoot growth. During the following season, lateral buds start growing. If the new lateral shoots outgrow the original terminal shoot year after year, a round-headed, or decurrent, tree will result. Excurrent trees tend to have strong apical control, resulting in upright trees with strong central leaders. Although some species tend toward one of these forms throughout their lives, all trees start out with excurrent traits as juveniles and most become more decurrent as they mature. Sweetgum, tulip tree, Liriodendron tulipifera, and most conifers are excurrent forms. In decurrent trees, the ability to maintain the strong central leader is lost, and a rounded form develops. Maturity and environmental conditions can also influence the growth form of trees. Many pines maintain a strong excurrent form throughout most of their lives, but develop a rounded top as they age. Limited access to sunlight, such as in forest understory conditions, can cause a normally rounded tree to grow upright. It is important to take a holistic view of tree growth and development. A delicate and changing balance of chemical signals controls the metabolic processes of photosynthesis, respiration, and all biological functions within a tree. Tree growth regulators impact each other and remain in a dynamic equilibrium. Environmental stimuli, including those caused by humans, trigger changes in regulator concentrations that, in turn, stimulate changes in resource allocation, which results in a response in the tree's growth or development. A system of defense. Trees cannot actively fight or move away from harm, but that does not leave them defenseless. Trees have a number of features that serve as protection: thick bark, thorns, leaf hairs, thick cuticles, and many others. In addition, certain cellular materials may resist decay or may be indigestible by insects. Another defense mechanism is the production of chemicals that resist insect feeding, pathogen infection, or decay. A developmental process unique to trees is the ability to compartmentalize, or wall off, decay. Compartmentalization is the process by which trees limit the spread of discoloration and decay. After a tree has been wounded, reactions are triggered that cause the tree to form boundaries around the wounded area. Alex Shigo proposed a model of this compartmentalization process, called CODIT, compartmentalization of decay in trees. In Shigo's model, the tree forms 4 barrier walls. Wall 1 resists vertical spread by plugging xylem vessels. Wall 2 resists inward spread by the more compact latewood cells and by depositing chemicals in these cells. Wall 3 inhibits lateral spread by activating ray cells to resist decay. These 3 walls form the reaction zone. Wall 4 is the next layer of wood to form after injury, new wood tissue, and it protects against the outward spread of decay. This is the barrier zone. Wall I is the weakest, and Wall 4 is the strongest barrier against decay. At times, the tree cannot resist the spread of aggressive decay fungi. It is fairly common for walls 1, 2, and 3 to fail, allowing decay to spread inside the tree, forming a hollow cavity. Wall 4 fails less commonly, but it can be breached by certain fungi. The barrier zone is strong chemically but weak structurally because the structural role of the parenchyma rays is discontinuous at Wall 4. The process that resists the spread of decay into new growth can also lead to shakes and cracks. Tropical trees and palms. Tropical trees. Tens of thousands of species of tropical trees have been identified, many in the rainforests of the world. The tremendous diversity among the species makes it very difficult to make general statements about their biology. Their anatomy varies greatly, probably much more than that of temperate species, and their physiology sometimes breaks the rules that temperate climate arborists have come to accept. The great variety of architecture, reproductive and defense strategies, and leaf morphology can challenge even scientists and educators. Tropical trees may lack annual growth rings because growth can be more or less continuous, or it may be highly regulated by wet and dry seasons. Many species do not experience dormancy except, sometimes, at the seed stage. Some species have extremely large foliage, flowers, or fruit. Some have a wide-spreading array of buttress roots at the trunk base that help support the tree and distribute mechanical stress. Some species produce roots from stems or branches above the ground, called aerial roots. Aerial roots are thought to have one or more of several functions, including support, serving as props, gas exchange, and propagation. New plants produced in this way are clones of the parent tree. Because of the favorable growing conditions in tropical climates, plants tend to have a rapid growth rate compared to those in temperate regions. In some ways, these favorable conditions have a forgiving effect, which aids in ease of transplanting, wound closure, and regrowth after injury. But just as with temperate species, tree managers must understand that when tropical trees are removed from their native ecology, challenges with growth and health are likely to occur. Palms. Palms are monocots and have more in common biologically with grasses than with hardwood trees. Palms do not have a cambium layer or growth rings of xylem. Instead, they have vascular bundles of phloem and xylem sheathed in exceptionally strong, fibrous tissues, embedded in a matrix of parenchyma cells and interspersed within the stem. The stem develops all the vascular bundles it requires for life behind the primary bud as it grows, and these vascular bundles typically retain their conductivity throughout the life of the plant. The stem is considered woody, although it differs anatomically from the wood of hardwood and conifer trees. Cells in the stem do thicken and strengthen as they age, and the stem is capable of storing starch in the parenchyma cells. Because the vascular tissues are dispersed throughout the stem, movement of water and minerals is not restricted to a few rings of xylem toward the outside of the trunk. With no cambium layer toward the periphery of the stem, there is no secondary growth, resulting in increased girth. These traits are thought to reduce the vulnerability from injury to the stem. However, palms do not have the ability to repair, grow over, wounds. Palms are not believed to have any process analogous to caudet, so wounds to the stem are permanent. At the same time, many palms are considered to be resistant to decay, probably as a result of the strong, hard sheathing fibers of the vascular bundles. Palms typically have only one apical meristem per stem. This primary bud is usually protected by the overlapping leaf bases of the emerging fronds. If this bud is lost due to injury or pest damage, the palm, or at least that stem, will die. During a palm's establishment period, the number and size of vascular bundles increases. Palms attain their maximum girth during this stage, mostly prior to beginning vertical elongation of the stem. Photosynthesis takes place in the fronds, which dominate the palm crown. Fronds are the leaves of palms, consisting of a blade, a petiole, and a leaf base. Leaves are produced sequentially. As the innermost, newest leaves emerge, the outer ones are pushed outward and down. Palms have the largest leaves of any trees, growing up to 60 feet, 18 meters, long in some species, with great variation in color, form, features, and thickness. The reproductive structures, flowers and fruit, emerge in or below the crown from the leaf axils, leaf scars, or nodes. Palm flowers are typically clustered into large aggregates of many small flowers. On some palms, these inflorescences are inconspicuous, but on others they can be quite showy. A few species of palms flower once in their life and then die. Because the inflorescences consist of many individual flowers, palms usually produce fruit in large numbers, although there is great variation in the fruit size among species. The root system of palms is also very different from that of hardwood trees. Like the stems, palm roots lack secondary growth, cambium meristem, and, other than the primary seedling root, they are adventitious developing from an area, typically at the base of the stem, called the root initiation zone. Most palm roots are in the upper 12 to 18 inches, 30 to 45 centimeters, of soil, and most are close to the stem, in a densely packed network called the root mat. It is not unusual for a portion of the root mat to be above ground. The roots of palms generally have a good capacity to resprout when cut, which, along with their adventitious nature and root initiation zone, contributes to their typical case of transplanting. Palms, like most woody plants, do form mycorrhizal associations.